welcome everybody. Welcome everybody to our Evenings at Whitney series here at the Lohmann Auditorium in person. Thanks all for coming on such a cold night. And also hello to people on Zoom. Welcome back. My name is Sandra Lösken. I'm a associate professor of chemistry here at the Whitney Lab and the University of Florida. And I'm your host tonight. So I have a really good speaker for you today. I'm very excited. But first, because there's some people on Zoom that might not know the Whitney Lab, we need to tell them a little bit about the Whitney Lab. As you all know here, the Whitney Lab was founded in 1974 and is a biomedical marine research institute of the University of Florida. It's composed of now 12 faculty, so a warm welcome to our new director, Veronica Hinman. There she is. As you see in several talks and also on our website, the Whitney Laboratory uses marine organisms for biological research that can be applied to human health, understanding of natural resources and ecosystems, and enable insight into basic biology, genomics, and evolution. You might see also on the website, the Whitney Lab trains scientists of all levels. So we're really proud of our kindergartner through university level to lifelong learners. And tonight, I'm really happy to see the whole community showing up in large numbers. Also, you know, the Whitney Laboratory features our Sea Turtle Hospital, which promotes ocean awareness and conservation through sea turtle rehabilitation, research, and education. And as you saw when you drove in here, the new building is coming up with the new Whitney Lab and the new Sea Turtle Hospital. So very exciting for this, and thanks for Mark Martindale is here for all the hard work on that new building. I think we're all excited to see this this year to be finished and moved in. So today I'm very excited to host a very close friend and distinguished professor from the University of North Carolina, Greensboro, Professor Dr. Nadja Cech. Nadja Cech is a fellow chemist, so don't be scared, just a little bit of chemistry. But she's focused on finding new drugs from natural sources. So she's trying to find new antibiotics, and her focus is looking into plant botanicals. Her work is funded by federal agencies, including the National Institutes of Health, and her research has informed FDA regulatory processes. Think about food supplements, teas, but also recreational drugs. So her path into science is a fascinating one, and she's currently writing her memoir. So I'm pretty sure there are some good vignettes tonight for us. So without further ado, welcome Nadja to the very cold Whitney Lab.
Hi, everyone. Can you hear me well? Oh, wonderful. So, Dr. Lösken, thank you so much for the introduction and for having me here. Thank you to everyone at Whitney for having me, and thank you to all of you who came out tonight to listen. And I'm really thrilled to be here presenting and to have the opportunity to share a story about science and life with all of you. So that's what I'm going to do today, and let's go ahead and jump into it. So the title of this talk is From the Wilderness to the Research Laboratory, my quest to uncover the mysterious healing properties of plants. And I'm going to talk mostly about one particular plant today, and you'll get to know it well throughout the talk. So I wanted to start out a little bit by talking about the scientific method, because this is something scientists bring up. I'm sure any of you who have even taken a middle school science course have heard about the scientific method, this idea that science involves a hypothesis, that we conduct experiments, that we get results and interpret those results, and then we can ask more questions, questions never end, and develop more hypotheses. And this idea is very central to the science curriculum, but a piece that's often missing in our science curriculum is where all of this comes from. We talk about it a lot as if it exists sort of in a vacuum, this amazing cyclic process that's completely objective and separate from human beings. But the reality is that humans are involved in every step of this process. We are the ones who come up with hypotheses, we're the ones that plan the experiments, we're the ones that importantly talk about what the results mean and what they mean for the greater community, and we're the ones who decide in the beginning what types of questions are worth asking. And so that's one of the reasons why, for me, it's very important to think about who is involved in doing science and to make sure that we have people with diverse perspectives and interests and questions that are doing the science making. And that's one of the things I love about my job at the University of North Carolina, Greensboro. This is my research group, or the research, I should say the group of scientists that I work with at the University of North Carolina, Greensboro. And it's always changing over time. In fact, one of my former group members, Joe Mengen, is in the audience tonight. He's actually doing a PhD at Whitney Labs now with Dr. Loskin. So it's wonderful to have people coming in and learning science and then having them move on and bring science elsewhere and continue on with the trajectory of that. So the results that I'm sharing today are all the result of the hard work of these people. I get to do the fun thing and come and talk about science with awesome audiences like all of you and get to be up here with a microphone on the stage. And they're the ones that are actually putting things in test tubes and running reactions over and over again and having experiments fail and then repeating them over and over again until they work. So I'm very grateful to them for doing all of that, and I really want to acknowledge them right at the beginning of this talk. What I'm going to be talking about today is the use of plants for medicine, and it's very likely the case that humans and also other species of animals, including chimps, and gorillas, have been using plants for medicinal properties pretty much as long as we have been around. There's evidence also that Neanderthals used plants for medicinal purposes. Some of the earliest writing of this is documented in what's called the Ebers Papyrus, which comes from ancient Egypt, circa 1500 BCE. And this photograph, this reconstructed image I'm sharing here is a fresco from the Theban tomb, which is around the same time period. And it's an image of an ophthalmologist treating a patient's eyes. And that would have most likely been done with plant as well as animal materials. So sort of before we had pharmaceutical medicines, we used what was in our environment around us for medicinal purposes, and we learned how to do that over many, many centuries of what I would call scientific exploration of seeing what worked and what didn't work. And that's where some of the knowledge we have today about medicinal practices comes from, is rooted in that ancient history. So plants, molecules from plants have very profound impacts on humans. And I'm going to ask all of you, can anybody think of an example of anything you know of from a plant that has an effect on people? Caffeine. Okay, I was hoping someone would say that. That's like such a great example, right? So sometimes people are like, is there really anything to this whole plant medicine thing? But then, of course, the idea that you can have an effect on your body from a plant molecule is hard to discount when you have to go without your coffee or your tea in the morning, right? Like, obviously there is an impact there. Any other examples that people can think of? Yeah. What was that? Digitalis, absolutely. So digitalis, the source of the cardiac glycosides that are one of the major treatments for congestive heart failure. Wonderful example. Didn't expect to get that one from you all. You're a very educated audience. Any other thoughts? Yeah. Echinacea. Okay, I heard someone say echinacea. I'm glad you did. I'm going to talk about echinacea today. That's the special plant, the secret plant. You already found it. Yeah, somebody else. Cannabinoids, right? Obviously, cannabinoids from marijuana, from hemp, CBD, have profound effects on the human physiology, right? So we could probably do this all night. There's a lot of examples. I wanted to highlight a couple that I think are really interesting right off the bat. One of them is this plant, Artemisia annua. Does anybody know what molecule comes from Artemisia annua that's used medicinally? You might not. It's this molecule here called artemisinin, and artemisinin is actually the most effective treatment for malaria. Malaria is one of the major killers worldwide, still today, and the history of therapy for malaria is that it's largely been based on molecules that have been isolated from plants. Quinine was the first one, and artemisinin has replaced it mostly as an even better therapeutic for malaria. And the woman who is credited with the, quotes, discovery of this molecule, which is kind of an interesting term, right? Because the molecule was there long before we came and discovered it. But the woman who's credited with developing the antimalarial therapy that relies on Artemisia is this woman, Tu Yu. And Tu Youyou was a scientist, retired now. She received the Nobel Prize in Physiology and Medicine in 2015. And she has a really interesting story, which is that she was hired in the 70s to run an institute to look for treatments for malaria. And she went back to several thousand-year-old Chinese texts and read the documentation in the texts about what plants traditional Chinese medicine doctors were using to treat fevers. Now, malaria, right, is caused by the Plasmodium parasite. So at the time that Artemisia, the plant, was being used by traditional Chinese doctors thousands of years ago, they didn't know about that parasite. They didn't know the mechanisms for malaria, but they discovered through experimentation and observation that when they gave this plant to patients who had fevers, that it helped. And so Tu Youyou relied on that information. She and her team took plants that had been used to treat fevers in traditional Chinese medicine. They tested them in experiments where they have cells growing in petri dishes, and they see if it can get rid of the malarial parasite that way, and then they tested it in animals, and then eventually they did clinical studies with humans. And one of the things I love about the story of Tu Youyou is that she insisted that she would be the first person to consume the isolated artemisinin that they took out of the plant before it went into clinical trials, because she wanted to make sure it would be safe before she was willing to give it to other people. So she's a really amazing scientist, and pretty inspiring story. This is yet another plant that's the source of an important molecule that's used medicinally. Does anybody recognize this one? Sandra recognizes it. Taxol, yes, so this is Taxus brevifolia, the Pacific yew tree from the Pacific Northwest, which is where I'm from. So I grew up around these trees. And these trees are a source of the molecule taxol, which today remains one of the really important therapeutics for ovarian and breast cancer. And when this became a therapeutic in the 1990s, it really changed the whole landscape for how people treated breast and ovarian cancer. And in many cases, survival rates for certain types of cancer went from, say, 30% to 70% survival rate with the introduction of this molecule. So it really has saved literally thousands and thousands of lives, mostly women's lives. And a big reason, actually the reason, that we have taxol is because of this man, Mansukh Wani, who was at the Research Triangle Institute doing research in the 1960s as part of a program by the National Cancer Institute. And the National Cancer Institute had started a program to look at flora and fauna, so plants from North America, and screen them for their ability to kill cancer cells, and then eventually test the successful ones in animal models. And the Taxus brevifolia was collected as part of that project, was one of 114,000 different samples that were collected. And it was one of 2 samples that became successful cancer drugs. And Mansukh Wani, I actually knew Dr. Wani towards the end of his life. When I was a young scientist, he was really inspiring to me. He spent a year and a half isolating the first half of a gram of taxol and 0.004% yield from the bark of yew tree. So this giant bag of yew bark showed up at his laboratory, and it had actually bounced around the US because nobody wanted to deal with it, and he said, I'll deal with it. And he and a group of scientists at the Research Triangle Institute worked with it for a very long time, and in fact, he was told that he should give up on this molecule. It was very complex. If you look at the structure, it's a very complicated structure. There's lots of what we call chiral centers in it, where it was hard to figure out the exact configuration of them. And Dr. Wani took on the challenge, and he was told to give up, but he kept working on it on evenings and weekends and eventually figured out the structure. So he was part of the team that really continued to champion this, and so that it eventually became a cancer drug. So I partly present all of that history because I now want to tell a little bit about the work that's gone on in my group, and I sort of wanted to give you the landscape of what it was that I was thinking about when I started as a young assistant professor at the University of North Carolina, Greensboro, thinking about using plants for medicine. And really, at the time, was thinking about two things. One was the traditional use of medicines, like in that picture from the Egyptian tomb, where we're talking about complex preparations of medicines that are being used and continue to be used in indigenous practices around the world and in what we call alternative medicine here in the United States. And then also the connection between that and the development of pharmaceutical drugs that we rely on, like the cardiac glycosides, like taxol, like artemisinin, many other examples that are isolated molecules that come from those plants. And all of that has to do with the relationship between humans and plants, which is so scientifically fascinating. So this is a photograph of me taken back in 2002. And this was basically within the first year and a half that I showed up at the University of North Carolina, Greensboro. And I'm standing there in front of an instrument known as a mass spectrometer. And what I thought I would do today, which is something I've never done before, is I want to share a little bit. I've been writing a book, as Sandra mentioned. I'm writing a memoir called Wild Chemistry, which is about my experiences studying medicinal plants and also doing research on them, and also my own background and how that informed my desire to do that. And I'm sort of very deep in the writing right now. I have to deliver the final draft at the end of this year. It's going to be published through Simon & Schuster. And so I have several excerpts from it that I wanted to share today, and I'm going to share one about a mass spectrometer. And like I said, I've never done this before, so I was actually, honestly, a little nervous to do it tonight. But you are the first people, aside from my editor and agent, to ever hear this. So I'm going to share a little bit. I'll do this now, and I'll do another little piece later. All right. I took the job because they promised me a mass spectrometer. My own mass spectrometer, a very large, very expensive scale capable of weighing very tiny things. That mass spectrometer would cost somewhere north of $100,000. It was a chance that I, a 23-year-old community college graduate with no high school diploma, could hardly imagine having been given to me. But it was right there in the offer letter, printed in Times New Roman 12-point font, signed in dark blue ink. The mass spectrometer would be mine. All I had to do was pick up and move to Greensboro, North Carolina, 2,834 miles from home, to a city where I knew not one soul. My life for a mass spectrometer. In the grand scheme of things, it seemed somehow a worthy exchange. So I picked up the few belongings I couldn't live without, my tabby cat, my chemistry books, a scraggly spider plant, loaded them into a U-Haul, and headed east. I pulled into Greensboro in the midst of a monsoon-type summer deluge. Squinting through the streaming windshield, I circled the identical brick buildings of the Lincoln Green apartment complex 3 times before finally identifying apartment 32A. I shut off the engine, and the rain stopped all at once, as if someone had reached into the sky and turned off a faucet. Unfolding myself from the driver's seat, I stepped into the steamy air, dizzy with the vertigo of dislocation. Kudzu-covered objects hovered at the edge of my vision, amorphous lumps disguised by the plant's snaking tendrils. Water dripped from every leaf. The kudzu seemed poised to engulf us all, the building, the U-Haul, my cat, and me. My new apartment was cavernous and uninspiring. White walls, beige carpeting, kitchen wall papered with hideous green stripes. Months ago, I'd chosen this place for what I thought was its best feature, an enormous walk-in closet. I had looked at the empty closet rods and imagined rows of neatly organized shoes and sweaters and dresses, items that I did not, in fact, own. Tonight, that closet would fulfill a different need. I dragged my futon inside, lay down on it, and slid the door closed behind me. To a girl who had lived out her childhood in yurts and cabins and tents, the closet felt just the right size. Awake on my lumpy futon, I stared up at the ceiling. My chest and stomach felt empty, internal organs somehow missing, and in their place, a yawning void. That void was familiar. I'd first experienced it in my college dorm room, plagued with insomnia, tortured by the easily audible voices of the customers in the Dairy Queen drive-thru at the end of the block. The void had followed me to grad school in Albuquerque. I'd lain awake there too, breathing in the exhaust fumes that seeped into my apartment through the swamp cooler, listening to the sound of the traffic on Lit Avenue, which was by turns deafening and quiet as the light at the intersection blinked from red to green, red to green. Now, with hot tears trickling down my cheeks and into my ears, I felt as if that void might suck me in and swallow me forever. It seemed that the easy fulfillment I had experienced as a child, connectedness to people and place, existed for me only in the mountains of Oregon. I imagined myself going to reclaim it, getting up out of this closet, putting on my pants, starting up the U-Haul and heading west. Tempting as that idea seemed, I stayed where I was. Somewhere in the Thermo Finnegan factory in Massachusetts, engineers were building me a mass spectrometer. I had plans for that machine. All right, well, thank you for clapping and thank you for listening. So now I want to tell you a little bit about what ended me up in that closet in the first place and about what we did with that machine. So this is a photo taken in 1983, actually, in Harlan, Oregon, which is in the coastal mountains of Oregon, and that's my family. The little girl there in the blue dress is me. My dad and my mom and my brother and sister are all there. We were standing in front of a whole pile of firewood that my dad had cut, and my parents in 1983 decided to go off grid and raise their kids in the Oregon wilderness. They were sort of the last wave of the hippie movement, and as you may be able to tell from this photo, and so that was how I got started, was living in this beautiful deciduous rainforest in Oregon. And I was also fortunate to do a lot of gardening. This is me again, sitting in front of my very first garden. And I loved my garden. I was very proud of it. As you can see, I took my nasturtium flowers and decorated my hat beautifully with them. And I spent a ton of time working with plants and getting to know plants. And I think that's what got me interested, of course, in the science of plants. And I also lived in a community when we, a few years later in 1985, my family decided to move down south to a commune in Southern Oregon. And part of the reason for that was that there was this alternative school there. And this is actually a photograph of us performing at our graduation ceremony at the alternative school and playing our recorders. And this is kind of what it was like to go to alternative school, which is also known as the hippie school, which was that there was a lot of time outside and really not a lot of school in hippie school. It was a great introduction. And I spent a lot of time in that type of environment. And then, that was 1986, and then sometime in 1993, I ended up meeting this guy who's circled in white in that photo. That's John Salinas, and he was my first chemistry teacher at Rogue Community College. You can see me, I was a proud chemistry student in front of the chemistry labs at Rogue Community College in this photo. I'm with my little sister there. There were kind of two things that I was really interested in. One of them was school, and the other was my family. And I noticed that actually all the photos that I pull up from this time period, I'm often with my sister. I'm there with her in the corner. We worked with John Salinas. I worked with John Salinas. I was hired to work with him to survey lakes in the Oregon wilderness and study the chemistry of lakes. And I just, of course, brought my sister along. That was just like what I did. And then John was the one who said to me, you know, Nadja, you should really major in chemistry. And I was like, really? And he's like, yeah, you can go to Southern Oregon University. It's right down the road. You can transfer there with your community college classes, and you could major in chemistry. But there was the problem of paying for that university education, and it turned out that my parents really did me a solid in that regard. This is a photograph of me with my brother and sister in front of a truck full of a plant. And I'm wondering if anyone wants to guess what the plant is. Somebody said it tonight earlier. It's echinacea. So that pink, it's really hard to see in the photo, I know, but that's me, and this is echinacea. So at the time, actually, this was 1994, my dad was working for a dietary supplement manufacturing company in their laboratory, and they were making medicinal preparations out of echinacea. And They needed more raw material, and so we spent 2.5 years growing this echinacea. It takes a long time to grow echinacea from seeds all the way to plants that you can harvest. And we sold this echinacea, and the money from selling this echinacea, my parents could have used it in many different ways, but they took all of it and paid for me to go to university. So it's really interesting that I ended up studying echinacea. It's not a coincidence, right, if we go back to that first slide on human beings and the scientific method, that echinacea and I have a long relationship together. I planted the seeds, I took care of those plants, I was out in the field with them, and they were the reason I was able to go to college. So when I went to university, I met the guy in this photo, Ed Espinosa, and this is me giving my first scientific talk at a conference in 1997 at the American Chemical Society in San Francisco, giving a talk about a protein called hemoglobin, which is shown in that photograph. And actually, this was an important watershed in my training because this was when I was first introduced to mass spectrometers. So when I met Ed Espinosa, he actually was affiliated with the university, but a scientist at the Forensic Laboratory down the hill from the university, the National Fish and Wildlife Forensics Lab. And Ed had the idea that if we could measure hemoglobin molecules in blood, we could use that to tell what species the blood came from. And that was actually a problem that was plaguing forensic scientists at the time. Ed was involved, we were at the Fish and Wildlife Forensics Lab, so we were involved in trying to solve crimes that involved animals. And often, if you're trying to solve a crime that involves an animal, so for example, someone has been poaching or someone has been trafficking animal products or things like that, often the evidence is blood, and often the question is, what animal did this blood come from? And blood pretty much looks like blood, almost regardless of what animal it comes from. And so, but it turns out that hemoglobin molecules, which are the proteins in blood that transport oxygen, differ slightly from one species to another. And so the idea was, can we develop a database where we use a mass spectrometer which can weigh molecules, and we weigh hemoglobin molecules from all different species of animals, and then use that to identify unknown samples and figure out what species they came from. And actually, Ed Espinosa was an amazing research mentor. So a lot of people who get involved in research have the opportunity to work with someone as an undergraduate student, as I did, who gets them excited about lab work. And Ed was amazing because on the first day I came in, he showed me a bag of evidence. It was little chips of wood inside a plastic bag. And he said, well, this comes from a crime scene. Someone was accused of killing a bald eagle, and we have no way to tell if this is bald eagle blood. We don't know. So the question was, can we develop a method that could be used to identify this evidence? And so I spent 1.5 years in the forensic laboratory analyzing blood samples from every kind of animal you could imagine, cougars and deer and bison and wood ducks and what have you, in front of a mass spectrometer day after day doing exactly the same thing. But it was interesting because I was going to be able to solve a crime if I could figure this out. And sure enough, Ed, after we had analyzed all these samples for 1.5 years, Ed was like, okay, it's time. You're going to be the one to do it. Analyze this evidence sample. And I knew what it should look like if it was bald eagle blood because I'd been analyzing bald eagle blood amongst all the other samples, and it was a very distinct pattern that we saw of the hemoglobin molecules from bald eagle blood. And so I took the sample into the mass spectrometer, and I took those wood chips and put them in the water and injected them in there, and sure enough, popping up on the screen was the data for bald eagle. And I was just like, okay, I'm a scientist now. This is amazing, right? Like, what are you going to do at that point? And I was also smitten with mass spectrometers. I was like, wow, this is really an amazing tool. You have something that looks the same, human blood or deer blood or bald eagle blood, and you put it in a mass spectrometer, and wow, you can answer a question like this. So it turned out then that I headed off to Albuquerque, New Mexico to work with this guy in the photo here, Chris Enke. He's shown with his graduate student, Rick Yost, and the two of them developed a new type of mass spectrometer that really revolutionized mass spectrometry, and they did that in the 1970s, as you may be able to tell from this photo. And I didn't actually end up working with Chris Enke until 1999, but he was at the time very respected as a mass spectrometrist. So that's actually what drew me from Oregon all the way to New Mexico. And even though my family was very important to me, as you see from those photos, I chose to go and study mass spectrometry in New Mexico with Chris Enke, and then eventually ended up at the University of North Carolina, Greensboro, which is where we began here back in 2002. So now I'm realizing that I have no idea how much time has passed. Is there? Oh, it's there in the back. Okay, we're great. We're great. We're on time. Okay, just checking in to make sure. All right. So now we're going to talk a little bit about the research that we did with this mass spectrometer once we got it, right? So that was all leading up to what it took to know how to use a mass spectrometer and to have one. So one of the questions that my group asked early on when we started working with mass spectrometers was, what chemical constituents, which molecules in the medicinal plant Echinacea are responsible for its effectiveness in treating or preventing infection? So at the time, I was quite convinced that Echinacea works to treat and prevent infections. That's how it's used in the alternative medicine community. There's a lot of people actually in Europe use it for that purpose as well. It's part of the pharmaceutical practice in Europe to treat common colds with Echinacea. And of course, I had spent a lot of effort growing all those plants that were going off to make Echinacea medicine. So it didn't really occur to me that they might not work, but there was a lot of disagreement as to how it worked and did it work. And so I thought this would be a good question for us to work on. In fact, I remember saying to my dad, we'll start with Echinacea and we'll get that figured out in a few months and then we'll move on to something else. That's what I figured would take a couple months. So we got some Echinacea plants. Actually, my dad shipped them to us, and these are a couple students in my laboratory working with those plants early on. And this is the type of data that we could get out of a mass spectrometer. So what it's called a chromatogram. And a chromatogram shows you a signal on the y-axis, and it shows you time on the x-axis. And essentially what's happening is that you have a mixture, such as in this case, an extract that comes from the Echinacea plant, and you've put it onto a column where it can get separated out based on the polarities of the molecules. And then over time, you see peaks, and every time you see a peak, it means that a molecule is being detected by the mass spectrometer. So each one of these different peaks is a different molecule, and based on when they come off the column, you can start to get a sense of what molecules they might be. And then there's additional information that we use here, which is, that's interesting. Wasn't the slide I thought we were going to have. Okay, hold on one second. We're moving forward for a second. Here we go. There's additional information you can use, which is we can look at the masses of the molecules. So remember I said mass spectrometers weigh molecules. So we basically take the molecules, and this is what the structure of some of the molecules in Echinacea looks like. We weigh it, and we break it into pieces, and we weigh the pieces, and based on the masses of those pieces, we can then kind of reconstruct the structure of the molecule. So in the case of Echinacea, the molecules that we were seeing on those peaks in the chromatogram were these ones. They're called alkylamides. If anybody has ever consumed Echinacea, you'll know that it has a very tingling sensation in your mouth when you consume it, sort of like Szechuan peppers. If anybody's ever consumed Szechuan peppers, they also actually have these same molecules in them, these alkylamides, and they stimulate the mechanosensitive neurons in your tongue, and it causes a numbing sensation, which is actually why this plant is also known as toothache plant. It's used to treat toothaches. And so we were detecting large quantities of alkylamides when we looked at the Echinacea plants, and I was really excited about those, partly because of my own background with the plant, which I'll mention to you in a moment. And so we came up with the hypothesis that these molecules from Echinacea have an effect on the immune system that helps prevent infection. And that was also based on other literature that was being done in other laboratories at the time. It was kind of the general thinking was that Echinacea was working through stimulating the immune system and preventing infections. And so we thought, okay, it must be that these alkylamides are the important molecules. And part of the reason also that I had that thinking was because my dad, Richo, who was the one who was working with the echinacea plants that we were growing, who first introduced me to echinacea, always had said that the stronger the tingle, the better the medicine for echinacea. So I had this anecdotal, information that people believed in the herbal community that the tinglier echinacea plants are the better ones. So we went out to try to explore this question. And I was involved in research that lasted way more than 3 months. That was a major underestimate. We really hardly could figure out how to, turn on the mass spectrometer in 3 months. I don't know what I was thinking. But anyways, we got going on this. And one of the things we did was to collect echinacea plants from all around the state of North Carolina. So this is one of the locations where we collected echinacea. This was in Asheville, North Carolina, outside of Asheville, in the city of Asheville in the mountains. And I'll show you a map in a minute about where we got all the different plants. And then we wanted to both measure the constituents in those plants and also look at their effects on the immune system based on the hypothesis we had. And so we needed to collaborate with somebody who knew something about the immune system, because I jumped into this knowing nothing about the immune system, which is actually, as it turns out, pretty much the most complicated biology you could possibly try to engage with. But fortunately, I connected with this colleague, Cynthia Wenner, who's at Bastyr University in Seattle, and she had been working also with echinacea. So I asked her if she would collaborate with us, and that's one of the wonderful things about science, is you get to meet people that you can collaborate with and do research with. And so what Cindy showed us how to do, I went out to her laboratory, and she showed us how to take echinacea extracts and add them to cells in petri dishes. And we worked with macrophage cells, which are some of the important immune cells that respond to an infection. They're the protector cells that will engulf pathogens that are coming into the body and destroy them. And we had the idea that we could add echinacea to those macrophage cells and then incubate it together and then look at what happens to the cells over time. And you can measure various response molecules in the cells, like what we call cytokines and chemokines, which signal to various pathways being activated within the cells, and get a sense of what's happening to those cells. So we're sort of doing two things in parallel. One is looking at the chemistry, and the second is looking at the biology of the cells, chemistry of the plant extracts, biology of the cells exposed to them, and trying to draw connections. And what we predicted was that the echinacea extracts would be able to stimulate the immune cells, and that would be correlated with the presence of those tingle molecules. So I was pretty sure that's what was going to happen. And as I mentioned, we collected echinacea from many different locations around North Carolina. So here's an actual map of that. And I don't want to sign into FaceTime. Don't. Interesting. Can't check that out. What's that? Yeah, we're good, right? Okay. Problem solved. Can run a mass spectrometer too. All right. So actually I can't, but Joe can. So here's the map of where we got our samples from, and here is one of the samples. Actually, this is sample 7. That's the photo of sample 7 being collected. And when we added it to the echinacea cells, it actually did not stimulate them, to the immune cells. We added echinacea to the immune cells. It did not stimulate them at all. It inhibited the production of this cytokine TNF-alpha. But when we took a sample from a different location, it induced the same cytokine. So we're basically seeing opposite effects of different plants from different locations. So let me ask you this. Let me turn this over to you for a minute. Why would you think that the same plant grown in two different places, one of them grown near the coast, another one grown in the mountains, might do totally opposite things? What do you think? Soil? Great. Okay, yeah, it could be the soil. Very good suggestion. Any other thoughts? Concentration of the molecules there, right? So what kind of factors would affect the concentration of the molecules? Is the plant under stress? What's the weather conditions? What part of the plant you're using, right? We have endless numbers of interesting hypotheses that we can generate about this that we could test. Any other thoughts? Hydration? Absolutely. What about the plants themselves? Are they all identical? No, right? They're all individuals. They're different genetically. People are growing them from different seed sources. They're probably all Echinacea purpurea, but they could be a different species if someone has misidentified it. So there's a lot of different factors that could be at play here, right? So when we saw these results from the biology, we weren't terribly surprised. In fact, it's what we were expecting, right? So we thought, okay, the chemistry must look really different for these plants. So we looked at the chemical profiles. We ran these chromatograms. We hooked them up to our mass spectrometer, and we started injecting all of our samples into the mass spectrometer. And what we were really surprised to see is that the profile we're seeing here is actually quite similar across the samples. So now granted, we can't detect everything in the samples, but the thing we were detecting was these alkylamides that I was so excited about. It's the same thing you detect when you consume Echinacea, and they were really similar in their profile across all these different plants. So that was unexpected. So we sat on that for a while, didn't really know what to make of it, and then there was sort of a buzz in the field around this idea of the relationship between plants and the plant microbiome. And it turns out that plants, like humans, have a microbiome. And there's a name for the organisms that live inside of plants. They're called endophytes, right? Phyte being based on plant, endo inside. So they're microorganisms, bacteria, primarily bacteria and fungi, that live asymptomatically within a plant, meaning it doesn't necessarily make the plant sick, but it's living there as part of the plant. In the same way that we have microbes living inside of our bodies, on our skin, inside our gut, and that sort of thing. And the photograph here is actually a photograph of an Echinacea root. So that little chunk that you see here in the middle is a piece of a root cut off of an Echinacea plant, and it's been sterilized on the outside and then placed on a petri dish. And what you see growing all around it is fungi and bacteria that are growing out of the root. So even if you think it's sterile, it is not, because there's organisms living inside of it. So we started to get interested. Somebody mentioned soil, and we started to get interested in organisms that are being introduced by the soil into plant roots that might be having an effect on how they interact with immune cells. In fact, we came up with a new hypothesis, which was that the immune stimulation we were seeing in the test tube, the samples that were stimulating the immune cells, perhaps they were doing that because of bacteria. And that made sense because it turns out, right, I mentioned that those macrophage cells that we're using, their job is to protect the body against bacteria. So you expose them to microbes and they go nuts. So what we were doing is taking Echinacea, and in fact, I will tell you that I had been cautioned to make sure our samples were totally sterile when we worked with them. Like, be really careful not to introduce any bacteria. So we kept everything so clean. We autoclaved everything, then we treated it with this special hydrogen peroxide solution, and we had gloves on, and we had all the glassware was so sterile, and all the tips were so sterile, and all the water was so sterile, and there was none of these. And I didn't realize that we were putting bacteria right into the samples right along with the Echinacea. So this is what happens when there's a piece of the experiment that you don't understand. That was the case for this experiment. And sure enough, what we ended up doing after much experimentation was we grew Echinacea in sterile chambers without any bacteria present or any microbes present. We grew the Echinacea in sterile chambers, and then we extracted these plants that had been grown in sterile chambers, and we looked at how macrophages cells responded to them. And so this graph on the top is just showing the signal that we see from the immune cells when they're exposed to the Echinacea, and so that's the signal here. And all these numbers are just different plants that were grown, individual plants that we grew in those boxes in the figure I showed you previously. And when we compared that to the amount of what's called lipopolysaccharide, which is a component of the bacterial cell wall that tells us how much bacteria is in the samples, remember, this was grown in a sterile environment, but there's still bacteria inside the plants, even if we sterilize from the environment. And what you see is a really strong correlation between which plants had a lot of bacteria in them and which ones had a really strong stimulatory effect on the immune cells. And Emily Britton was the person in my group who did a lot of this work. And she was the one who developed the experiment we could use to measure the content of the bacterial cell wall component. Turns out we couldn't detect it with the mass spectrometer. It was too big, so we needed a different experiment. And that was why it wasn't showing up in our data, the chromatograms I showed you earlier. And we got a really strong correlation between these two. So the LPS that comes from bacteria was really strongly correlated with the signal we saw in the immune cells. And what's really interesting in this project, to me anyways, the most interesting part is that somebody mentioned soil earlier today. The microbes that live in soil are really different depending on what soil environment you're in. And it turns out that which microbes you'll find inside of a plant, the plant microbiome is very strongly linked to the soil microbiome. So it's not necessarily the case that you can assume, oh, echinacea is echinacea. It must have certain bacteria living in it all the time because they like echinacea. Not really. It's more that echinacea will have bacteria in it that reflect the environment that it was grown in. And so if you grew a carrot in an environment next to an echinacea plant, those two roots would probably have very similar bacteria. And if you grew it in a different environment, you would probably see very different bacteria. And that's what's being shown in this photo, is just some experiments that we did where we went out into the field and collected echinacea plants and looked at how the bacteria component differed in different environments. And we saw very different bacteria profiles depending on the environment where we grew those plants. So just to summarize here, what we saw, I didn't actually go into the data on this part of it, but I wanted to share it, was that the alkylamides from echinacea, those tingle molecules, were actually still important. It turns out that they, when you add them to macrophages that have already been stimulated, they suppress the response of those macrophages. And so this is kind of consistent with if you have a cold or a flu and you've got symptoms from that, and you want to take something to suppress that inflammation, which we often would think of using something like, say, Tylenol or Advil. But echinacea actually, on a much less potent level, it will do somewhat the same thing, at least with these experiments we're doing in Petri dishes. It's hard to sometimes extrapolate that to humans. And the bacteria from the echinacea, we're doing exactly the opposite. So if you add that to unstimulated cells, you get a stimulation of the response. And so that's why we were seeing, when we harvested plants from different locations, opposite effects. Some of them, the alkylamides were dominating and we were seeing a suppressive effect on the immune cells, and in other cases, the bacteria were dominating and we were seeing stimulation. What this means for humans is tough to really tease out. A lot of people use echinacea. There have been many clinical studies on it. Some of them have shown it to be effective. Others have shown it to be ineffective. No studies have really shown it to be particularly toxic or dangerous, which is good. So if you are using echinacea, I'm not a doctor, but the literature suggests that that's not a problem. And if it's helping you, awesome. I've thought a lot about what this means to have spent, as it turned out, more than a decade studying echinacea. We studied other things at the time as well, but it took us a long time to figure all this out. And what I've come to is thinking a lot about the relationship between humans and plants. And part of what got me thinking about that was getting back to gardening. This is a photograph of my two kids, Nova and Jaden, at the Dunleaf Community Garden, which is right down the road from where we lived for many years in a neighborhood in Greensboro, North Carolina. And I'm showing here a watermelon that we grew in the garden and how proud they are of that. And when I go back to this question of does echinacea work, I always think about the garden. And I think about how echinacea was used before we started trying to bottle it up and sell it in, GNC and health food stores. And the echinacea plant was really sacred to indigenous peoples that lived in the plains of North America, which is where it is native. So before we came in and developed North America, you would find echinacea growing wild in these giant plains of pink flowers. And the indigenous peoples used it to treat toothaches. They used it to treat snake bites, though there's no evidence that it actually does work for that. And they used it also as a spiritual plant. So I wanted to read you a little bit of my thinking about echinacea here as my last little excerpt. So here's me in the garden. That is not echinacea, but it's another plant in the garden. All right. Since 1943, the decade when Watson and Crick, having stolen data from Rosalind Franklin, published the helical structure of DNA, we scientists have been obsessed with genes. Genes fit comfortably in our reductionist model. We love the idea of peering into a cell and making predictions just by reading off A's, C's, G's, and T's. All life laid out in code. More and more, though, science points to our genetic code not as a prognosticator of our future, but as a series of blueprints that map out many possible futures. What matters is not just genes, but gene expression. As we interact with our environment, the genes within us receive constant signals, as if an operator is sitting at a switchboard and flicking them on and off. On, off, on, off, on, off. It's happening all the time at an impossibly rapid rate in every one of your cells. Even though I will never be able to carry out experiments complicated enough to prove it, I've come to believe that my time in the garden is sending the right messages to my genetic switchboard. As I step off the concrete and into the garden, I can feel something coiled tight inside me relax. I've planted echinacea in the Dunleith garden. It's taken years to fully take root, but this summer it's positively bursting with powder pink blooms. I sink down on my knees and bring my face close to them. A swallowtail butterfly has landed on one of the flowers. She unwinds her proboscis and slides it easily down into the powdery depths. Fluttering her wings with a gentle flit-flit rhythm, she sips away with what I can only describe as butterfly delight. Does echinacea work? I ask myself for the 40 millionth time. Liquid amber swells up in my chest, and it occurs to me that maybe I've been thinking about this all wrong. Whether or not I dig it up and chew on its roots, this echinacea plant is healing me all the same. Sitting here beside it, I feel better already. All right, so there's some spoilers for my memoir. I hope you all read it. I want to end by just acknowledging again my amazing research group that I'm so lucky to work with, and funding that we've had to do the work I described here and many other projects from the National Institutes of Health. Also wanted to make a shout out to the Dunleith Community Garden. That has been an amazing community of people that I've worked with, and many students come and work at the garden too, so we have a great connection between our chemists and our plants, and I am convinced that our microbiomes are being shaped by eating plants that are grown in a community garden, but again, that is fully a hypothesis that has yet to be tested. And finally, I just wanted to also thank my husband who's sitting in the audience today. He came with me to Florida, which he very rarely comes with me when I'm on a science trip, and it's really great to have him today. This is a photo of our family when we went on research leave while we were at the university. And you know, in my earlier reading, I was sharing some of the trepidation I had when I first came to Greensboro and left my family behind, but I was fortunate to be able to build a new community and connect with Gavin thanks to the fact that I came to North Carolina, and that has been a really wonderful thing. So, you know, home is where we choose it to be, and that's also one of the messages that I share in my writing, and I'm happy to be here at your home today, and I would love to turn things over now to all of you to ask whatever questions you might have.